Hola. Gracias por tu atención y su bienvenida. And that's all I got, so. Maybe next year, I'm working on it, so thank you so much. I'm really excited to be here, and I really want to talk to you about what is going on in our field, what is happening with biotechnology. And it's really incredible, some of the pieces, and we've started to think about this next century here. The 21st century is our biotechnology century, much as a lot of the 20th century was the digital century, and I really think we're just still about halfway through getting started with that. We're now in this biotechnology century. And what does that mean? Well, part of what that means is we're starting to treat DNA and reading DNA like code. And that's what a lot of my career has been about. So I've actually been somebody who's been a computer scientist, a biophysicist, a molecular biologist, and I've worked at a lot of different companies. I've gone to a lot of different schools to try to put some of this together. A lot of my current projects are educational projects where we're trying to make sure that we're reaching more and more people and getting the word out about what's going on with all of these technologies. And the place where that really comes together is looking at DNA and how we are now able to read DNA, and that it's cheaper and cheaper to go ahead and look at that and do that. And this is kind of what I've worked on for the last 20 years, and this is what it's been costing. And you can actually see that price going down at a Moore's Law rate here at the first part of the 21st century, and that's not bad. Moore's Law is actually fantastic. That means you're decreasing the price by 1.5, 2 times every year. But a little company that I worked for got acquired, and we had a little technology that kind of worked, and then it worked better and better and better, and we started making that get cheaper at a 5x a year, year after year. So it's gotten remarkably cheaper. It's gone from $3 billion to read the first human genome to somewhere around $60 today, and that's in less than 20 years. So this is where we're at. And people sometimes ask, well, that's great. We're in this $1,000 genome era. We're getting down to the $10 genome. What does that really mean for us? What are the things that we can do with this technology? One of the first big things is we're learning how to do pharmacogenomics. And it's actually not very hard. What's going on here is each of us, whenever we take a drug from a doctor, we process it a little bit differently because we've got genes in our liver that go ahead and express themselves differently and sometimes make things go faster or slower, sometimes make a drug not work at all. And so this is a chance for us to go ahead and read those genes and know beforehand, before a doctor prescribes a drug, that this will work differently in different people. And actually doing the testing for this is now very, very cheap. But the reason we don't have this as a widespread thing, because it costs about $7 to look at these 30 genes. We're still not very good at electronic health records. This is one of the few places where computers are actually holding us back from the things we want to do. So, and once you have electronic health records, you've got to have not just the record of all the tests, but a way for the doctor to read this, to be able to look at it and know what's going on with it. And this becomes a bigger and bigger deal. Doctors actually don't read blood tests. They don't look at pictures of blood cells under a microscope. They send it off to a lab. The lab sends it back and it says, blood test, okay. And we want to do the same thing with this. The ones that I've seen that work well go ahead and say, for cardiac disease, or for this kind of cancer treatment, or for someone who's being treated for depression, for mental illness, here are the drugs in the green category. These work pretty well. Here are the ones in the yellow category. There might be a problem, but you're okay taking them if you know what's going on. Here are the ones in the red category. Don't take these if you've got an alternative. And this is a good step forward, and we're getting better and better at doing this. The other thing that we've gotten better at is being able to use this technology to look at an unborn baby and see, is there any kind of a problem with the pregnancy? And this is something that replaces an old technology called amniocentesis. How many people have ever had to do amniocentesis? Excellent. I see a couple of men who have done amniocentesis. Come talk to me afterwards. That might have been an unnecessary procedure for you, sir. And I kid with people. I raised my hand. I have twin boys, and whenever I went in with their mother, and we went ahead and did the needle in the side, and she winced and I winced at the same time, and then they had to repeat it on the other side. You know, those are separate amniotic sacs. They had to do it twice. And so I'm holding her hand, and I'm trying to make her laugh. And the doctor says, "Good God, man, don't make her laugh. You'll move the babies, and we've got a needle." I'm just like, ah. I mean, that's all I knew what to do. She had to go through the hard stuff. I was just trying to be funny. So now we don't have to do it anymore. Instead of the needle in the belly, we do a needle in the arm. We take a regular blood sample. We send it off to the lab. The lab looks in the blood sample, finds the DNA in there. 95% of that DNA is from the mom. 5% from the baby. We can look at that 5% and say, everything looks good, or there might be a problem. We're getting rid of this whole amniocentesis in almost all of the developed world. In the developing world, we're not even starting there, because any place you can draw a blood sample and you've got access to DHL or FedEx, you can get it to this lab. So people do this now all over. And that's not where it stops. It turns out it's kind of happy to have DNA in your blood from a baby. The sad thing that we found, every once in a while somebody will have DNA in their blood because they've got cancer. While we were actually doing this testing and figuring out how this worked, we found a couple of cases where we said, this is the mom's DNA, here's the baby's DNA, here's something else. And I was like, that looks a lot like what I've seen for colorectal cancer. And we were able to call the doctor and intervene and do something about this, and it had a happy ending. The mother was okay, the baby was okay, the cancer got taken care of. Now this is available more and more as a test that can be used. If you've got really cheap genomes, you can read the DNA of something, you understand the cancer system's biology, then you have this simple blood test for cancer, and we're calling it a liquid biopsy. And the idea is you take a little blood, you look in there, and sometimes whenever a tumor is just starting to form, when it's smaller than the point of a pin, and you can't even see it on an X-ray, any kind of imaging, you can go ahead and pick it up because it's releasing DNA into the blood. It's bad at being a tumor. Those cells break open. And so we can go ahead and see the signature, and we can catch somebody who's got cancer, not at stage 4 or 5, not at 2 or 3, not at stage 1, at stage 0, when it's just starting to be established, and a small treatment of chemotherapy, something like that, will do some good. Now, right now, this is available as a prognostic test. This is something that people will do because they can go ahead and they have cancer, and we're trying to track what's going on with it. Do these drugs still work or not? We hope to have this available as a diagnostic test, so that whether you have cancer or not, you're just a member of the population, we can take a blood test every year and look and make sure you don't have cancer. And if this is successful, and we hope to know in the next few years, then that means no more mammograms, no more trips to the crusher, okay? No more colonoscopies. Both of these things are so unpleasant, I had to put cartoons up, I'm just saying. They don't let you put up real pictures of that. So this is kind of where we're at. And this is, again, another step forward using this technology. Now, it doesn't always have to be about cancer or human DNA. Sometimes we can do stuff with food. We can go ahead, and here's a company that goes ahead and it sequences food. It's like a Consumer Reports. They went out and they bought 100 different brands of hot dogs, and they did the DNA sequence for all of them. How many people like salchichas? Yeah? I am so sorry for what I'm about to show you. I apologize from the bottom of my heart. Lo siento. So this is what happens whenever you run the DNA on 100 different brands of hot dogs. But the first thing they found, about 14% of these were problematic. What does problematic mean here? It means that all beef hot dog? Not so much. Maybe a little chicken, maybe a little pork, maybe a little bit of something else. So that's a thing. Then the second big thing they found in their test was they tested also vegetarian hot dogs. I was like, who eats a vegetarian hot dog? But okay. They tested the vegetarian hot dogs. 10% of those actually had meat in them. I'm thinking, you're eating the vegetarian hot dog, you're like, this is really good. It's the 10%. No. Oh, you're not going to be applauding when I show you the last thing. Here's the last one. 2% of the samples had human DNA. I love that sound. Oh, what's going on? People see that, and this is what they think. They think of Raymond late at night in the carniceria, working, going ahead, working over the grinder. So tired. And then the boss comes in the morning like, where's Raymond? I don't know, but look at all the work he did. Let's go ahead and stuff some sausages and get him out there. Not that bad. It's not even Wendy's where like somebody chops off a thumb and you find it in the chili. No, it's Raymond's working late at night in the carniceria and he forgets his hairnet, and so a little bit of hair falls in, or he clips the side of his thumb. It's not a whole thumb. It's just a little bit of fingernail or something. It's 2% of these samples had traces of human DNA, but that's kind of bad enough. It's a hygiene issue at this time. And then what this means is this technology actually lets us go and kind of use this as a microscope to examine our food products, to examine our environment, and see in a new way what has been there. And so this is sort of a wake-up call for industries. There are people who are adopting these technologies to watch you guys. And so you might want to go ahead and get there first. And we're seeing this a lot with not just DNA testing, but with AI and data analysis, with the use of drones to fly over big plantations, things like that, and find out what's going on, not at the edges, but in the center. All of these exponential technologies are a new way to do transparency. And I think that that's almost always good, depending on who's doing it to whom, right? You may not want it done to you. So we're starting to see more and more of this. We're also seeing this big explosion of consumer genomics, this ability where you're buying access to your own genomic data. And this is a huge deal because it's not just a medical test that you go to a doctor to get, it's something you get and you own and that you can do something with. And it gives you a chance to sort of touch the future. More and more of these companies are coming up. We actually track 250 companies in the United States alone, 400 companies worldwide that are doing this and offering some kind of genomic test, usually not a medical test. And some of that, I think, is fantastic. Some of it, I'm a little iffy on. Look at some of these categories. Look at those big red question marks. Matchmaking? You can actually scientifically show people who have really different immune systems tend to be attracted to each other through the scent of pheromones to promote mating outside of your kin group. But that is not a great first date, right? So I'm a little iffy on that. There's some scientific support, but who's going to go to do that test? I mean, online dating is bad enough. So trying to do that one. There's a couple of things where you can actually go and say, here's a woman, here's a man. We can look and say, your baby will probably look like this. But again, do you want to show up at a first date with that picture? So child talent is another one that I really worry about. They've taken some of the testing that we've done among extreme athletes, people who are Olympic caliber, that is really used and useful for going ahead and training people to see what somebody's muscle reactions are, what's their capacity for inspiration and going ahead and breathing out their VO2 max, what's their recovery time, and how can you go ahead and adjust their training to get another 1 or 2% performance out. But using that on kids to say, you're good at soccer and you're not great. I always say, if you want to find out if your kid is a good runner, don't get a genetic test, get a stopwatch, right? It's just a lot easier and it's better on everybody. So I'm a little iffy on some of those uses, but most of them, I think, are this great way to touch the future and do something fantastic with it. And you guys have seen Origin is here. They do consumer genomic tests, and they've offered a free test to everybody where you get to look at 6 different variants. This is a fantastic way to go ahead and do some interesting things. So if you haven't had a chance to take that and complete that, go ahead and give it a try. See what you think. And then as we go forward, the technology for this just gets better and better and better. The machine that I helped design, which is like as big as a Volkswagen and cost $1 million, is being replaced by a little thing that's about this big and costs $1,000, $10,000. And it can go ahead and run your genome. It doesn't even have a power supply. It plugs into the USB port on a computer. I just love that thing. Give us a few more years and we'll have something like that that is fully independent, where you can use it to go out and do environmental DNA testing, that kind of thing. I have this dream that I'll be able to go on a nature hike with my twins, you know? And they'll say, "Daddy, what kind of a tree is that?" And I'll say, "Well, son, that's a yucca plant." Because I know biology for little bitty things, but anything that you can actually see within a microscope, I have no idea. They'll be like, you know, Daddy, what kind of a plant is that?" I'll be like, "Oh, that's an agave plant." What do you use that for?" "You're too young, don't worry about it." "Daddy, what kind of an animal is that?" "Well, go catch it and let's see." And they'll come back, "Daddy, I didn't catch it, but it bit me." And I'm like, "Well, let me see." Okay, you don't have rabies, so we're good. "And Daddy, I got poo all over me." "Well, we can see what it ate. Let's go ahead and look at that." It's like, "Oh, that was a brown striped squirrel, and it was eating other brown striped squirrels. Let's get out of here. I think the hike's over." And this is the kind of thing we'll be able to do. And then we'll be going home, right? And they always want to stop and eat on the way home. And whenever I'm back in Silicon Valley, they always want to stop at the IKEA to eat. Here, they would want to stop. There would be somebody who's selling tacos on the side of the road, which I dearly love. They'll stop over and see, and they'll go like, "Let's get some tacos." I'll go, "Let me see." Okay, the cow icon came up, so we're good. Everything looks good. And then they'll eat about half of what we get. We'll go back. They'll fall asleep. I'll eat the other half. That's what I do. And then they're going to wake up, and they're going to say, "Who ate all of our leftovers? There's an empty plate. Let's get Daddy's DNA reader, and we're going to see who ate out of that plate." And it was Daddy. "Oh no, he's not our daddy." You gotta watch out. Hey, I'm a geneticist. I check day 1. This one's mine, this one's mine. We're okay. But we're really at a point where we can use that DNA to do things and trace people and test things. And what does that start to look like for all of us, right? It becomes really interesting. Maybe that matchmaking thing is not such a bad idea after all. Maybe we could actually make that happen. Maybe I could have a little thing where I could walk around the audience and I could go ahead and put in my parameters for, a good date. A good spouse is what I meant to say, Mom. And it would come up and it would start beeping, and I could go, like, wait, he's got a Y chromosome, sorry, but you're a good-looking man. And keep going. It's like, oh, early death from cancer. That's not good, because I'm going to have the same thing. I'm just saying. So one of us has got to take care of the other. Stand up, because I think we have to go on a date. This is my girlfriend. I get to embarrass her like this. So, a hand for that. She's going to make me pay for that later. So this is kind of our future, and really we will be using this to see paternity. You can buy DNA paternity tests in the drugstore now. We're out of Q-tips. Would you pick up that in a DNA paternity test? Ooh. You can go ahead and find people who haven't actually taken a test because one of their relatives, 3 or 4 spaces removed, has. And this is what happened when they caught the Golden State Killer recently. One of the most prolific serial killers ever. He was so good at doing it because he had been a policeman and knew what to do to dodge, but didn't anticipate that almost 30, 40 years later, there would be a technology where they could find him through his family. These are the kind of things. Most of this is good. Some of it's a little scary. Probably the era of DNA privacy is already over, because the idea of some scanner that I could wave around, that's very real. What do we do with that? What do we do with the next part of the technology, where you're not just reading DNA, but you're writing it? These are some of my favorite acronyms. These are the things that are very important technologies. Don't worry about the small print. The big deal is what it actually does. CRISPR is a way to do genetic engineering with these programmable scissors for DNA. It cuts where you tell it to cut. CAR-T is a way to reformat your immunity, to tell your immune system to go after different things and be much better at what it does. And we've got the first genetically engineered drugs that do this released just this last year. RNAi is a way, it's a mute button for DNA where we can actually turn things down so that the cells don't express certain genes without going in and reprogramming the cells themselves, but maybe just spraying this on a plant, saying, don't turn on these genes. Maybe spraying it on some bugs, and the genes that they would turn on would be the ones that keep them alive, and so they just die out. It's kind of a scary form of genetic engineering that happens without having to genetically engineer anything. And then SCNT is a way to change one cell into another. And just in the last 2 weeks, some things have happened that I want to show you. But first, let's talk a little bit more about CRISPR and the fact that all of these things taken together are not just acronyms for scientists and technologists, but they're things that are going to change your life and the lives of your family and the way medicine works and the way all of our industry works. Any kind of manufacturing, anything in this room that we build for a technical civilization in the next 20 years will probably be built by biology because of these technologies. The next time you see one of these acronyms in a newspaper article, in a magazine, please read that article. You will then know more about it than 99% of the people on the planet, and you need to know about it. So CRISPR, we already talked, it's like a Swiss Army knife, a toolkit for changing genes. It's something that works cheaply, it's very targeted, it's improvable, so it gets better and better, and it's worked in every kind of cell we've ever used it on, from yeast and algae and little single-celled organisms to all sorts of animals and plants, including humans and including human fetuses. And we'll talk about that. The big thing about it is it's so much cheaper than what has come before. So 5-6 years ago, I do a gene editing experiment, and it takes me 6 months and $20,000. Now it takes me 2 days and $200. And this is how quickly you can move with this new technology. The first place we're really going to see use of this is in industrial biology, reprogramming single cells to do what we want them to do, to make things that we want them to make. We've actually been doing this for 20,000 years because we've been using yeast to make bread, to make beer. Just we weren't very good at reprogramming it. We had to go ahead and breed it over generations. Now we can just go ahead and make pinpoint changes. This is letting us go ahead and do things like make new medicines, reprogramming E. coli to make medicines to treat diabetics, or make food and food additives, different things that are hard to find in nature. There's a good chance, there's about a 1 in 4 chance that the next thing you taste that is vanilla might not actually be from a vanilla plant. It might not even be a synthetic vanilla from chemistry. It might be grown up in a microorganism. And so we're seeing more and more of this as it gets to a bigger and bigger scale where you don't just make a little bit in a laboratory or a couple of liters, but you're making big tanker trucks full, 200,000 liters. This becomes something we use to make bioplastics, biofuels. As one example of some of the companies that are doing this, There's so many people working on this. They're making fibers, different things for, weaving cloth. They're making fuels, like we talked about. But the most interesting ones are the ones on the bottom of that chart. They're making all the tools. Every time there's a gold rush, don't grab your pan and pan gold. Go ahead and start selling people pickaxes. And we're in the middle of a genetic engineering gold rush, and those are the people who are making the picks and shovels for it. And it's very interesting, the pieces that are going on. As one example I did want to share, this company, FredSense, they actually changed a bacteria into an arsenic detector. They took a naturally occurring little arsenic sensor, put it in a bacteria, and made it so whenever it sensed the arsenic, it didn't just swim away from it. It changed its electrical field, so you can pick this up within a couple of minutes. This is kind of amazing. They were doing this work. They were students doing a summer project, and they were doing this with some funding from a big oil company who had been drilling. They said, hey, we've made something that's 100 times faster, cheaper, and more sensitive than the way you guys tell if you have caused any kind of environmental damage. And the oil company said, no, thank you. We don't really want to know that quickly if we've got that kind of a problem. We're fine with the old way. And so they said, who could possibly use this? Detroit said, yes, please. Here we have a whole city whose water system was having high levels of arsenic. If they had known about it, if different people had known about it. And so they're now making this sensor and 5 other ones for city water supplies. At some point, this will be something that fits on your faucet and lights up whenever you go ahead and want to see if your water's okay. This is how fast this is moving, and it started as a student project. The next thing that we see being a big deal is digital agriculture. How do we use this technology to change the way that we grow our food? This is from a little study that I did for the Inter-American Development Bank. They had been looking at what are all the different technologies that are being used, which ones are on this exponential curve, and which ones are appropriate in different regions and are actually at a really good timing point in the exponential curve. And we identified so many technologies and starred the ones that we thought deserved special attention, and did some studies with examples. And almost everybody wants to talk about engineering crops and GMOs or other kinds of ways to gene edit our plants. And I think that's really important, but I think the thing that may be even more important that we will all see even sooner than some of the long-range pieces there is this, cellular agriculture. This is the idea that you could go ahead and get meat without killing an animal to do it. And that's really kind of profound if you think about it, not just because you don't harm the animal, but because all of the trouble that goes into growing an animal for months or years and then killing it and throwing away about half of it, all of the water and feed, all the damage to the environment from growing that and from the waste goes away if you are growing these cells in a tank and harvesting them very precisely there. I show this picture to my kids and I say, we can go ahead and have bacon without hurting the pig, and they say, Daddy, we're pretty sure you're hurting that pig right there. That is a big chunk out of that pig. And I say, it's a bad diagram. We don't cut the chunk out of the pig anymore. We do a thing now with a hypodermic. We go ahead there like a shot. I'm like, exactly, like a shot, like getting a shot. They're like, we don't like the pig getting a shot either. I'm like, well, do you want the pig to get a shot and you can still have bacon, or do you want a dead pig? They're like, okay, no, let's give the pig a shot. That sounds good. So we pull the cells out of the pig, we grow them in a bioreactor, which is actually fairly easy to do, but you then have to organize them into sheets and muscles, and you've got to put in vasculature, veins, so that you supply the cells with blood and you circulate it in this thing. And so you're growing real meat, real tissue in there, and you actually end up putting it on a Pilates machine. You're exercising your muscle cells in this bioreactor so that you can take them out and then not get a flabby piece of bacon. You can get some really good. You can get a piece of steak is the idea. And it's not even genetic engineering. People talk about genetic engineering, yes, but the idea here is that we're just growing it in a different place. Now, we can genetically engineer it too. We can turn down the cholesterol. We can turn up the omega-3 fats, all the things that you would get. We could make that bacon like salmon for you. We could do some different things. If you're a little sensitive to the milk proteins, we can turn some of those down. We can do all those things. And people look at this and they say, this sounds like science fiction. We don't think you're really doing that with the pig. Here's the thing. We've got so many companies that are not just working on this, but producing results. Right now, in the United States, there are over 1,000 restaurants that are serving Impossible Burgers. Those are made with plant proteins and a genetically engineered heme, blood, really, that makes it sizzle. They're pretty good. Get them at medium. Medium rare, they're not quite there yet. But it's not bad. And this is just going to get better and better. The one that you have this year will be the worst one you ever have, because it'll be better next year and better next year. At some point, that cow, Bessie, will have a Facebook page, and she's going to live to a ripe old age, and people are going to be like, Bessie's delicious, and we just eat burgers that were made from Bessie all the time. This is where we're going with the technology. Now, I want to take a little bit of time to really look at what it means to reprogram humans. What happens with this technology when we start using that in humans for medicine and for reproduction? And Vivi and I had some really interesting conversations about this, and she had wanted to talk about cloning, and I said, cloning is kind of now old. We've had people doing cloning of mammals. She's like, yes, but the first human who's cloned, and I'm like, don't tell anybody, but I think we've already got the first human that's cloned. And she's like, really? I was like, we could invite them, but then which one shows up, and it's embarrassing. But I actually wanted to invite these guys, cute little fuzzy mice, but mice not as you know them. These are mice that started out as cells and dishes. So they're not actually just cloned, but these mice don't have a dad. They are a result of in vitro gametogenesis, which is a fancy way of saying the sperm and egg that made them actually came from cells that were turned into sperm and egg. And those cells both came from female mice. So these are mice that have two mothers. And there are several implications here. For one thing, once we start being able to do this in humans, the idea of infertility kind of goes away. If somebody can't make sperm on their own, we can still make one that is genetically theirs. If someone can't make an egg on their own, we can still make one that's genetically theirs. And it really doesn't matter which sex it comes from. And if that doesn't blow your mind, you're not paying really good attention. So that's the first thing. The other thing is all the things that we're now doing in humans. It turns out it is so much simpler to apply all of these technologies. Manipulating single cells, changing the genetic code in a cell on the scale of not just a single organism, but a fetus. I've got 10.5 trillion cells, maybe 10.7 trillion. I'm just saying. A little baby has got much less, and in fact, there's a stage at which they have even much less than that. These are pictures of embryos, of babies, what we call them our blastocysts. 3 days after conception, sperm and egg come together, divides once, twice, 3 times, you get 8 cells. You can do a lot with 8 cells. You can take one of those cells, and people hear me say that, and there was this one woman who stood up and was like, no, don't take the cell. I'm like, no, it's okay. The baby just grows more cells. It's not like the baby shows up without an arm. It's good. You get 7 cells, it still grows into 10 trillion cells. But you can take that cell and you can look at it and read all the DNA and see, is it going to be okay? Is it not? Is there something? If this isn't the embryo that you wanted, we could put it to the side. And is that okay to do? What if we're saying, we've already had 5 boys, we want a girl? You could keep going until you find a girl, and you could have that one, implant that one. What if you have mom and dad have cystic fibrosis? They are carriers for it. They want to have a baby that doesn't. You could go ahead and search through the embryos until you find one that doesn't, one that's not even a carrier. So it never happens again in the rest of your family's life in future generations. We can do that. We can look and say, here's somebody who's at a much higher risk of getting breast cancer or ovarian cancer. We could select one that's not. This is done now. This is called prenatal genomic selection. And people are doing this, although not openly. It's still not considered ethical. It's not available commercially in most places. But people are doing this. And I know a little bit about it just because whenever I had my babies, this is how they started. Those are my twin boys. This is how they grew up. They turned out okay, I'm just saying. So this is the kind of thing you look at. This is the new family portrait for the 21st century. You use in vitro fertilization. You have access to human cells, and it's not just fate, what is born. You can pick. You can even alter. So we're at the point now where we know CRISPR is still a new technology, but we're very good at using it to knock out genes, turn genes off. There are certain genes, it turns out, we are better off without. Some people who are just alive today, by the luck of the draw, have non-working copies of these genes from mom and dad. If you have non-working copies of CCR5 in the middle of that list, you are immune to HIV. You cannot get HIV. That's a nice trick. And we can turn those genes off in an embryo. And this has now been done in China in an experiment where they didn't raise the kids all the way up to birth, as far as we know. FUT2 is something, I happen to have a non-working copy of FUT2 from my mom and from my dad. That means that I am immune to noroviruses. I cannot get food poisoning. I like that. That means I can go on a cruise ship anytime I want, right? Everybody else is like, oh, I'm like, give me some more pate, right? I can walk into a kindergarten and all the kids are running around and the teacher's like, the kids are kind of sick, be careful. I'm like, I don't care. And I won't get it. This is not bad. And we can engineer this. We can engineer strong bones. We can make somebody who will never get diabetes, no matter what their blood sugar does, they'll never get diabetes. Now, we can do it. It's being done. Here's an example of a study that was done in the US where they were able to go ahead and eliminate a congenital heart defect gene in one of these blastocysts. And again, they didn't carry it all the way to term. They've eliminated that CCR5 and made HIV immune blastocysts. But what happens next? We can do this. Should we do this? What should our ethics around this be? Who will be in control of this? It won't be the people themselves if we're doing these new procedures on fetuses. Not until they've grown up, and then it's a little late to reverse it. Maybe not for everything, but for a lot of things. Will the parents decide? Will the doctors? Will the government? My thing that I always say is just the last place we should put the control of this is in the hands of the politicians. They're the furthest away from the problem. But I don't know. And it bugs me. As an engineer, I wanted a black and white answer on this, and there's just not one. There's not a best group that you can say, in all cases, this is the group that should decide. This is the procedure where we should share control. I think what's going to happen on our planet is every different country, there are going to be 183 different legislatures and groups of people and societies that understand a little bit about what's going on and make some of this forbidden and some of it mandatory, and they just pass it on like that. This is where we're going to go. So the last bit, the little bit of time I got left to talk to you, I wanted to talk to you about kind of what happens next. What do you do whenever you realize that you now have access to what's potentially the most dangerous technology that the human race has ever seen? How do you get a good ending from that? My answer is you put it into the hands of 5-year-olds as quickly as you can. And I'm really serious about this. And this is something we're really doing. 10 years ago, we founded a not-for-profit called BioCurious. It's a community lab, a hackerspace for biotech, the first one in the world. And the way it really works is it's a gym membership, but for science. So people come in, they pay $100 a month, they take a safety class, they train on some equipment. We give them access to $3 million worth of high-tech biotech equipment. And we have students and artists and inventors and entrepreneurs coming in and doing amazing bits of work. We do a lot of things in there. This is a picture of it the first day that it opened almost 10 years ago. This is a picture of one of the classes that we did. The first day people come in and they make a PCR machine, a copy machine for DNA so you can have enough DNA to do things with. And they actually solder it themselves. And then the next day they extract their own DNA, put it in the copy machine, make more copies of it so they can send it off and get it sequenced. And here a young woman is looking at her BRCA1 gene, a gene that tells whether you're going to have a higher or lower chance of breast cancer and ovarian cancer. She's got a good reading on this. We got a nice accolade from Fast Company for the #4 most innovative ed company in the world, which we didn't really consider ourselves all about education or even really a company. We're a not-for-profit. We don't have a full-time employee. We were, though, happy to see so many people coming to do what they had been working on in their garage, working on in our lab. More and more people who were working on these things, inventing this open PCR machine that you saw people making, inventing some other projects that were just amazing. We've had so many companies come through. We're getting close to having $200 million of follow-on funding for companies that were founded or did some of their first experiments at BioCurious. And that's great for an organization that doesn't have a single full-time person working for it. One of the companies I'm most proud of that did their first experiments there, Miraculous. And Jorge Soto is going to be up here, and he's going to tell you about what happens in Miraculous. So I don't want to steal his story, but I do like saying the beginning of their story happened at our little community lab. And I hope to see another 20 companies that are as wonderful and as successful as the one that he started there. We've seen a lot of different people copy this model and go out and build a biohackerspace, a community lab, in their own places. And again, this is something I'm so happy about, and to see this spring up all around the world. If you want to see if there's one near you, go to DIYbio.org, DIYbio.org, and you can go ahead and see a list and also a forum where people are saying, I want to start something, will you help? These hackerspaces are new places for innovation to happen, new places for everyone from companies to nations to foster innovation. These are some of my favorite ones in the world, and really this is a list that's always growing and changing. The things that you can do with this. I still have my own garage lab. Back at home, I do experiments with my kids. Sometimes we launch companies out of there. And I love that, but I don't think everybody should have to have a garage lab to do this. I don't think everybody should have to have access to a biohacker space to do this. I think everybody should be able to do that. Like when I was a kid, I had a chemistry set that I used. And I would always ask my sister, I was like, come here, taste this, see what happens. And she would go, Mom, and my mom would go, are you making her taste things out of the chemistry set again? Like, it's NaCl, that means it's salt, it's okay. She goes, don't do it anymore, I'm gonna take it away. I want to have that same conversation with my kids. Are you hacking the family dog? Don't do that anymore, or you don't get a biology kit next year. We actually now have biology kits. This company, Amino Labs, makes a little, like an easy bake oven for genetic engineering. And it's something where you can actually alter bacteria so their colors are different. You can program what colors you want to be. You can program how it smells. You can even program how it tastes, although it says, do not taste the genetically engineered bacteria. Do not feed this to your sister. I like that. And then I would just want to end with one last bit. How do we bring this about? How do we get this stuff to happen for us, for our future? We need to have more people working on this, more entrepreneurs, ecosystems of companies, not just in Silicon Valley, all over the world, and especially here. More people doing this. And there are government ways to do it, but what's the best way to make this happen? So I was at a conference at MIT. It was a 10th anniversary of the first biohacker space being founded. People from all over the world had come, and somebody put up on a bulletin board this note: help and used equipment to set up a community lab in Mexico. Let's collaborate. And then Patty Rodile at gmail.com put her address up. So I hunted her down at the conference. I said, tell me about what you're doing. What are you trying to do? And she told me about the things that she was doing. She mentioned how she's really impressed with Mexico City, has got a place called Gene Garage. I was like, I've been there. They do a nice job. She goes, yeah, but we need to have things like that in more places. I was like, well, what about Tech de Monterrey? She's like, everybody says Tech de Monterrey. You know, what about the other places? She goes, Yucatan, they've got this thing called Plasmid Lab. We want to do something like that. We want to go ahead and do this. They're calling it micros. They're trying to put it together. And I said, man, there ought to be a way that we could help you do that. And so that's why I'm actually sharing her email address with everybody. If there's somebody who wants to collaborate, if there's somebody who's working on a lab, let me ask, how many people here are educators? Anybody who's doing that, please stand up for me. Any educators? Any students here who are interested in maybe making a biohacker lab or being a bio-entrepreneur? Okay, stand up. You guys stand up. Everybody look around and look at each other and find each other so you can talk to each other and talk to me and make something happen and contact Patty and do something. Seriously, this is how it happens. This is how we start. And I just wanted to say, too, I'm so happy to be involved with that community and be involved with you guys, and I want to give back a little bit because it's given me so much, and I'm so happy to be here. So I'm actually, whenever I get home, I'm getting one of these open PCR machines in a kit. They've got to put it together, and I'm going to send it to Patty, and you guys let me know where we should send the next one and start helping each other do that, please. That's my homework to you. That's what I ask you to do. So we're going to try to start a revolution. For Che and Einstein, you put them together, this is what you get. You get more tools in more hands, and more people are working on this, and you create this incredible future. This is where we're at. I'm hoping you guys will come, join us in this future. Find me here. Thank you so much.